Alrighty, so we're going to talk about Unit 5. There are 115 slides, and it covers 3 chapters from our textbook. It's basically genetics. So the first one, just title. So there's sexual reproduction, there's asexual reproduction. Sexual reproduction is when we have a male and a female parts, and then asexual is when the parent cell makes an exact duplicate of itself. So we're mostly going to talk about sexual reproduction because that's what happens in humans. Asexual reproduction happens in different kinds of organisms. It's technically what mitosis is. And then like bacteria and other stuff reproduce asexually, and then it gives you an exact replica. The risk of asexual reproduction is if it is vulnerable to something, or the organism is vulnerable to something. All the organisms are vulnerable to that something for the most part. In sexual reproduction, what it gives us is genetic variation, which then leads into something is better than the other, and so then we have survival of the fittest, which is Unit 7. But that's the why do we reproduce sexually, because we get genetic variation from that. And so this just talks about how we get the different parts that we need in order to go through it. So we'll talk a bit about meiosis as we go through. And so whenever we have sexual reproduction, say we're going to make an egg or we're going to make some sperm, we go through meiosis so that we can have half the amount of chromosomes. So for humans, we would have 23 instead of 46. And so you get 23 from mom in an egg, and 23 from dad in a sperm, and then they'd come together, they'd make 46 in the zygote or the embryo, and then it would replicate, mitosis, differentiation, and then it becomes an organism. When we talk about cells in our body, we refer to them in different ways, sort of fancy science ways. Somatic is a word that means body cells, like skin cells, hair cells, everything in your body except for the egg or the sperm. Genes are the little sections or segments of a chromosome that code for a specific trait, and alleles are the different forms of the gene, like you have a blonde hair allele and a brown hair allele, and then the dominant allele would be the one that's shown. Just use that in a sentence. Then this is just showing you how on given homologous chromosomes that there are segments that are homologous, not necessarily the same, but they code for the same thing, like hair color, or do you make lactose or lactase or whatever. Just another picture. And then when we make egg and sperm, we call those gametes. And then just some more vocab down there of immature reproductive cells. So before they're fully an egg, or before they are fully a sperm, there's like little steps that they go to in order to develop. And so up until that point, we call them different stuff. And then meiosis gives us eggs or sperm. The egg and the sperm, or the gametes, are haploid. We refer to that as N. It is half the number of the diploid number, which we usually refer to as 2N. And then if you take 1N and 1N and you add them together, you get 2N. Alrighty, so then just a little bit more about that. Meiosis is kind of the same as mitosis, but it has 2 divisions instead of just 1 division, and then there's several other things that make it slightly different. But kind of the steps that it goes through and sort of what it kind of looks like is pretty similar. So they're called meiosis 1 and meiosis 2. And so in meiosis 1, that is when homologous chromosomes, so an X and an X, which I always did this in class, so the X and X are these separated from each other. And then in meiosis 2, these are sister chromatids, and so then they would go like this, and then you'd have another set. Over here. Where's my camera? And so then you'd end up with half the amount in 4 things instead of the same amount in 2 things. One of the most important things that happens before mitosis and meiosis is the replication of the genetic material. So we have a double set and then it splits and then it splits again. So this is just a really generalized overview of meiosis. And then whenever we have an egg and a sperm and it comes together, that's fertilization, and then the product of that union we call a zygote, is the first little bit. And then this is just talking about how it cuts them in half, and then they become 4 haploids. Then just another little simple overview. And then some pictures. One of the things to look at in this, which we'll talk about in a bit, is crossing over. It happens during prophase 1. And so the little bits of the homologous chromosomes twist together and sort of exchange that little bit of genetic material, and then those chromosomes are different than they were originally, and then it gives us a whole bunch of different possibilities. And then just meiosis 2. And then this talks about crossing over. And then this is one of the biggest sources of genetic variation. It's why we have millions and millions and millions of possibilities of what babies could be born, along with the law of how they line up during metaphase, and then where they sort of, you get what you get and you don't throw a fit when it goes into the 4 egg or the 4 sperm. And then this is just talking about it, that it occurs during condensation, which means that when they go from chromatin to chromosomes, we talked about that in the last unit. And then just a little figure. And then this talks about how we calculate the genetic possibility. So this for us is the formula, I guess. It would be 2 to the n. N is the haploid number. So for humans, 2 to the 23rd is 8 million possibilities. So it would take, I'd have to have 8 million babies to have genetically a twin. Could I have one before that? Yes, probably not. But anyway, I'm not willing to do that. I'm sorry. But that's what that number's showing you. And then males give us sperm, and then females give us eggs. In males, they develop into 4 fully mature functioning sperm. In females, of the 4, we only develop 1 of the eggs, and then the other 3 are polar bodies. They break down and they get sort of recycled in the pieces and parts of the things. But in a female, we can't use all the eggs that we make now, really. And we only make a quarter of what we could. So our body is not going to expend the energy to make hundreds and thousands of eggs, because we won't use them. So we have evolved to only develop 1 of them. It takes less energy. We're not going to use them anyway. And then we are choosy. Remember we talked about the video with sexual selection, that we have put a lot of work and energy into the eggs. So like birds and like bighorn sheep and different stuff like that, the males are real competitive because they have to impress the females. That's an evolution thing. But it's also was a funny lesson in class. So this just shows you how the males develop into 4. And then if we were to look at that picture, that previous picture, and I were to put it up with eggs, there'd be 1 big one and 3 little small ones. This is just talking about fertilization again. And then a picture of lots and lots and lots of sperm around 1 egg. I talked about this in class. The instant that the first sperm reaches and sort of breaches that membrane into the egg, a chemical reaction, a sort of sealing of the egg happens, and so then another sperm can't get in. So it's a pretty cool thing that happens right at fertilization. And then this is just talking about some BRCA genes and what they have shown us and given us. Again, the BRCA genes are the breast cancer genes, but it talks about fixing and not being able to fix, and that's why the BRCA gene is bad, because if there's a mutation in the BRCA gene, then we can develop things that we shouldn't have developed, and that's a bigger picture than maybe just breast cancer. And then chapter 14, moving along. And so this is talking about just sort of an example of genes and how we can get traits from them. And then a cute little picture with some cute little babies. And then just talking about human skin color, a little picture over on the side. And so there are ways that we can chart and track and draw pictures and try and predict and all the different stuff that comes with genetics. So this one's talking about pedigrees, and we can put your relatives or a famous family's relatives or whatever and try and track the traits through pedigrees. And so this is just talking a little bit more about that. And then I'll give you a little picture. And so we practice these a little bit in class. And so males are squares, females are round. How they're connected, the little thing, the ones that come off the bottom, if it's filled in, if it's not filled in. Usually, if it's filled in, it's showing the trait that the pedigree is tracking. Sometimes you'll see questions and it'll say the colored in squares are the normal offspring or whatever. So if you have questions that involve a pedigree, just make sure that you read it real close. And then this is just an example of something we could track. It's called polydactyly. And this always reminds you of The Princess Bride because of the six-fingered man. But anyway. And then just a pedigree. And so if we look at this, there's a few things that we can look at and figure out. The first thing is it dominant or recessive? And is it a sex-linked trait? And so if it's dominant, it usually shows up in every generation. And so we would say this looks like it might be a dominant trait. If we look a little bit closer on line 2, then you might look up to 1, and then it has 2 non-polydactyly parents gave birth to a polydactyly baby. Now then you might say, oh, that might look like that. And then we're thinking about recessive. And so then it could be either, but we'll figure out what it is. And then is it sex-linked or is it not? So if it were sex-linked, more males than females would have it. On this, there's slightly more males than females, but there's still quite a bit of females that have it. And so then we would probably guess that it's not sex-linked. And so then there's just another example of Huntington's disease. Huntington's disease is a really interesting disorder because there are no symptoms and there are no even hints that you have it until like 35, 40, 45 years old. And the part about that is you've already probably had kids, so you've passed on the gene when you had no idea that you had it. So then now we know that things run in families, and so then they can say, oh, Huntington's disease runs in our family, and then you have to sort of maybe do genetic counseling and stuff like that. And then this is just talking about how we get different traits. There's several different ways. This talks about polygenic. Poly means many, genic means genes. And then epigenetics is things that when the environment can affect your genes and the genes of the next generation, or maybe decisions that you made, or actions that or behaviors that you did. And so then these are the big ones that we talk about. So autosomal dominant, autosomal recessive, X-linked recessive, and X-linked dominant, which means is it on just any old chromosome 1 through 22, or is it on the X or the Y? There are a small amount on the Y, but there's way more on the X, so we don't talk very much about Y-linked. We mostly just talk about X-linked. And then this is just some examples, and some more examples, and some more examples, and some more examples. And then one of the things about mutations is it always sounds bad. Usually it sounds bad. And so, but mutations are how we get changes that may lead to evolution. And so it's not always bad, but sometimes it is. And so this just talks about recessives and heterozygotes and different advantages. So we'll talk about these. And so an allele is inherited in an autosomal dominant if the trait that specifies appears in heterozygous people. So if we are big T, little t, and we see it, it's dominant. If it's big T, obviously we're going to see it. If it's little t, then you wouldn't see it. And so this is just talking about some examples of autosomal, not sex-linked, dominant. If it's present, you're going to see it. And so achondroplasia is a type of dwarfism. Huntington's disease I talked a bit about. And then progeria is the sort of aging in reverse disease. Maybe Google it. It's pretty interesting. And then this is talking about autosomal recessives. So albino or Tay-Sachs we talked about in class quite a bit. And then X-linked is on the X, but usually gets shown in males. And then that's because the males have one X, and if it's faulty, it shows up. Females can get X-linked disorders, but both of their X's have to be faulty because they have that other X that might have a dominant good allele on it. So just some examples of those. And then this is a little test. Hopefully you can see those. If you can't, the one on the left says 29. And then it says there's a 7 in this. I don't see the 7. And then the other one's an 8. And then this is just a pedigree of with hemophilia. And now we'll talk about chapter 13. And so this is just a review of some vocabulary. Just some examples of where scientists have figured out where exact genes are located on given chromosomes. Pretty interesting. And then we have genotypes, which is the letters. So you might be big T, little t. Those are genotypes. Two bigs or two littles are homozygous. A big and a little is heterozygous. Heterozygous are also sometimes called hybrids. And then they give us a trait or what we see or the physical observable traits. We call those phenotypes. And so then this talks a little bit about what I already mentioned. And then some examples. This is Mendel did Punnett squares with big P versus big P or little p, and then he got 100% big P little p's. So he crossed a purple flower and a white flower and he got 100% purple flowers. Then he took those purple flowers, those big P little p purple flowers, crossed them with each other, and then he got a 3-to-1 ratio. And so that just talks about the segregation. Then this is talking about the segregation of how the big T and the big T little t or little t, they segregate from each other. And so they are, they can give us different traits depending on the one that we happen to get. And so then this just talks about a little bit of that. And then the examples with the purples and the whites. And then Punnett squares are used to determine probability. It's a mathematical thing. It's not what we're going to get. It's what are the chances that we're going to get that. And then just a picture. We'll go through these. And so it's just showing you how you fill in a Punnett square. Hopefully you know that by now. And then a test cross is a word that AP likes to ask about. A test cross is what you do if you're not sure. Say we have a purple flower and we don't know if it's big P or big P little p, then we can do a test cross. It's sort of like draw a Punnett square on a piece of paper and then say if we get these colored flowers, then it was big P. If we get this colored flowers or this proportion of colored flowers, then it was big P little p. And then monohybrid is when you cross one gene or one trait against one trait. So flower color or seed color or anything, one thing. And then we talked about the P generation, F1, F2. And so Mendel, for the example that we just talked about, the P generation was big P crossed with little p. And then the F1s were the babies of that offspring. And so the 100% big P little p's. And then if you take a big P little p and cross it with a big P little p, and then you get big P, or big P, little p, big P, little p for a 3-to-1 ratio, those babies are the F2s. And so then this just talks, or just is the writing of that. And then the traits that Mendel studied. And then Mendel talked about the law of segregation, which is when the alleles line up, you sort of get what you get, and then they separate, and then a little bit goes to that egg, and then a little bit goes to that egg, or a little bit goes to that pollen, and a little bit goes to that pollen. And then just some pictures. And then a dihybrid cross is two traits at the same time. We got a 3-to-1 ratio in the F2 generation of a monohybrid cross. If we do the same thing of a heterozygote and a heterozygote, you get a 3-to-1 for a monohybrid. If we do a heterozygote and a heterozygote, then you get a 9-to-3 to 3-to-1 ratio. 9 is the dominant. 3 is dominant recessive. The other 3 is recessive dominant, and then 1 is the recessive traits. And so then this just shows you how to fill in a dihybrid cross and determine the gametes from a given parent. And then the law of independent assortment is when we make the gametes at the end of meiosis 2, the sister chromatids or the alleles independently go wherever they want to go. And then this just talks about a little bit about crossing over, which we talked about earlier. And then the likelihood of something crossing over is higher if they're further apart. If they're close, they usually don't cross over and switch and get separated from each other. And so sometimes they're called linked because they're so close on that chromosome that they're not going to bend over and like wrap around and then exchange. And so they usually get inherited together. And then these are going to talk about exceptions. And so codominance is when you have two alleles that are codominant, and if they are both present, they both show up, like blood type, like A and B are codominant. And so if you have both, they both show up. Multiple alleles means there's more than just two alleles, like big T, little t, or big S, little s. There's three. And again, blood type is one thing that has that. You have A, you have B, and you have O. O is recessive to A and B. A and B are codominant to each other, but both dominant over O. And just some examples. And then incomplete dominance means you have kind of a dominant and kind of a recessive, but they kind of blend in the middle. And so we know that they are incompletely dominant because we have an intermediate phenotype. And so for this snapdragon color, you'd end up with a pink flower instead of a red or a white. Just a picture. And then this just shows you how that works. And then epistasis is when something in this genetic and this genetic affect each other. And so you might make brown fur color, but you lack the gene to express it. And so it comes out unexpressed, so then it looks white or whatever that might be. So it just gives you an example there. And then a cute little doggies. And then how we get those on a dihybrid cross. And then pleiotropic is a gene that gives us multiple traits from it. One example is marfans. They are really tall. They have really long limbs, weirdly long fingers. And so that would be one of those. And then this is just talking about how the environment can affect. And so just click through these. It's just a little bit of background and vocabulary. And so we can mess with genetics sometimes. A lot of times they do it in plants. But our genes can get methylated, or so they can get like a methyl tag or an acetyl tag on it, or taken off, or put up. Usually it's put on, and then it either tightens up the DNA or loosens up the DNA, and then it gets expressed or maybe not expressed, and that's epigenetics. And then this is an environmental factor that would affect like the color of the fur. You can probably imagine snow or not snow would give us a different, or heat or not heat would give us a different expression, because that's what those bunnies, in this case, need to survive. And then this gives us an example of methylation and stress, which is the mental illness links to genetics is super interesting, but a rabbit hole we just don't have time to talk about. But if you're bored and you want to look it up, it's pretty cool. And then this is just talking about things with variation of traits. It is usually more than one gene coming together. And so like height or hair color or skin color or eye color, anything that's on like a continuum can give us, is usually more than one gene coming together and acting and to give us sort of one trait. And so then this just talks about that a little bit with some vocab. And then a couple graphs, just because these are females have not as perfect, because it's just a little bit different between the males and females. And then this gives us most guys are about 70 inches tall, which is like, I don't know what that is, 5'10" or something like that. And not too many of them are super tall. Not too many of them are super short. So it's just a funny little difference. And then sometimes we get weird stuff because of mutations. We know that. And then this is campylodactyly. Just your fingers are sort of bent, and then they are bent. It's not like you're holding them there. That's just what they look like. And then there are different ways that we can get mutations, so we'll go through those. Duplication is a little section is duplicated, and then you have a little extra. Sometimes it is taken out, and then you're missing it. Sometimes it is flipped, and then it's the order of the nucleotide sequences is opposite now. So then you get a different trait because you have a different instruction. And then translocation is when you take a bit of one and move it to the end of a completely different chromosome. And weirdly, on these, it happens in the same way in different people. So a cool example at the bottom is a little chunk of 8 and a little chunk of 14 switcheroo, and it results in what's called Burkitt's lymphoma. It's not very common, but it's common enough that they gave it a name, and 8 and 14 are always involved. That's weird. So just sort of an example. And then we care about things that make us stronger. Evolution cares about things that make us stronger. And so most of the mutations or the changes or different stuff like that has resulted in making us more fit. And then this just talks about how X's and Y's developed. Pretty cool. And then sometimes we have extras or not enough. And then it gives us different, oftentimes negative disorders. And so polyploidy, like we had haploid and diploid. Polyploid means there's more than one. And then an aneuploidy, we have an extra chromosome or an extra or not enough individual chromosomes. Polyploid is we have a whole extra set of 23, but that doesn't happen in humans. We would just not be born. But sometimes it happens in other things. And so when it is supposed to separate in making the eggs or the sperm, so during meiosis 2, they should separate, but sometimes they don't. And so if the two get stuck together and they travel instead of separating, then that would have too many, and the one would come over. And then the sort of the oddball sort of doesn't have any, and then it would get the one from maybe the egg, and then it ends up with 47 or maybe 45. And so this is just a picture to show you how that works. And then Down syndrome is the one of the most common ones because it survives the disorder is in individuals that survive. And so there's an extra number 21. And so we're maybe not super familiar, but you can probably recognize because Down syndrome results in pretty recognizable physical characteristics in different sorts of things. There seems to be a correlation with advanced maternal age. And so that's just a thing that if you have babies and you're a little bit older, they'll watch and they'll test for, because there's a test that's available for Down syndrome. And then this is just a picture of that. So this would be a boy with Down syndrome. So if you look, there's an X and there's a Y, and then there's an extra little number 21. And then just a little picture. And then some other examples. And so there's females, there's males, and they have different things with the X's and the Y's. We have too many X's, we have not enough X's, we have too many Y's, we have not enough Y's. And then it just gives us different things. They're pretty cool. If you wanted to Google them and check them out, they're interesting. But just know that there's differences. And then I talked about this a little bit earlier. When a disorder runs in your family, now people talk about things. And so you might be aware of it. And so if something runs in your family, you might do a pedigree or do genetic analysis or get your chromosomes analyzed and make a picture, a little karyotype, and then make an educated decision whether you want to have a baby or maybe do in vitro fertilization or do pre-implantation genetic diagnosis and say, I want the eggs that don't have cystic fibrosis implanted. It's pretty cool that now we can do that. But that's why we do all these tests, is to give potential parents an educated decision. Cool. And then this is just sort of the technology behind that. And it's just talks about taking a little piece of tissue or taking a little bit of fluid and testing it to see if there's any genetic abnormalities. Sometimes they do it after the fertilization and after a woman is pregnant. And then technology is also going towards doing it even before fertilization or before implantation takes place. And so this is just talking about that part is you can test the embryos and then only implant the embryos that are healthy, which is cool. A little scary, but pretty cool. So that's it.